Breathing and exchange of gases. As you have read earlier, oxygen is utilized by organism to indirectly break down simple molecules like glucose, amino acids, fatty acids to derive energy to perform various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reactions. It is therefore evident that O2 has to be continuously provided to the cells and CO2 produced by cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in following section of this chapter. 17.1 Respiratory organs. Mechanism of breathing vary among different groups of animals depending mainly on their habitats and level of organization. Lower invertebrates like sponges, coelenterate, flatworms, etc. exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms use their moist cuticle and insect have network of tubes, tracheal tubes to transport atmospheric air within the body. A special vascularized structure called gills, bronchial respiration (GB) are used by most of the aquatic arthropods and mollusks, where vascularized bags like lungs, pulmonary respiration are used by the terrestrial forms for exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration also. Human respiratory system. It leads to we have pair of external nostril above opening out above the upper lips. It leads to nasal chamber through the nasal passage. The nasal chamber open into pharynx, a portion of which is common passage for food and air. The pharynx opens through the larynx region into trachea. Larynx is cartilaginous box which help in sound production, hence are called sound box. During swallowing, glottis can be covered by thin elastic cartilaginous flap called epiglottis to prevent the entry of food into larynx. Trachea is a straight tube extending up to mid-thoracic cavity which divides at the level of fifth thoracic vertebra into right and left primary bronchi. Each bronchi undergoes repeated divisions to form secondary and tertiary bronchia and bronchioles, ending up in very three terminal bronchioles. The trachea, primary, secondary, and tertiary bronchioles and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchioles gives rise to a number of very thin, irregular walled and vascularized bag-like structure called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs. We have two lungs which are covered by double layer pleura with pleural fluid between them. It reduces friction on lung surface. The outer pleural membrane is in close contact with thoracic membrane, thoracic inner lining, whereas the inner pleural membrane is in contact with lung surface. The part starting with external nostril up to the terminal bronchi bronchioles constitute the conducting part, whereas the alveoli and the ducts form the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clear it from foreign particles, humidifies and also bring the air to body temperature. Exchange part is set of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in thoracic chamber which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by vertebral column DV, ventrally by sternum VS Virendra Sehwag, laterally by ribs LR, and on lower side by dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in volume of thoracic cavity will be reflected in lungs pulmonary cavity. Such an arrangement is essential for breathing as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First, breathing or pulmonary ventilation by which atmospheric air is drawn in and CO2-rich alveolar air is released out. Second, diffusion of gases O2 and CO2 across alveolar membrane. Third, transport of gases by the blood. Fourth, diffusion of O2 and CO2 between blood and tissues. Fifth, utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Now, 17.2 mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between lungs and the atmosphere. Inspiration can occur if the pressure within the lungs intrapulmonary pressure is less than atmospheric pressure, that is, there is a negative pressure in lungs with respect to atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm is a specialized set of muscles, external and internal intercostal between the ribs, helping generation of such gradients. Inspiration is initiated by contraction of diaphragm, which increases the volume of thoracic chamber in the anterior-posterior axis. The contraction of external intercostal muscle lifts up the ribs and sternum, causing increase in volume of thoracic chamber in dorsoventral axis. The overall increase in thoracic volume causes similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. Relaxation of the diaphragm and intercostal muscles return the diaphragm and sternum to their normal position and reduce the thoracic volume and thereby the pulmonary volume. This leads to increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the body. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in a breathing movement can be estimated by using a spirometer, which help in clinical assessment of pulmonary functions. Now, respiratory volume and capacities. Now, tidal volume of air inspired or expired during normal respiration, it is approximately 500 ml. That is, healthy man can inspire or expire approximately 6000 to 8000 ml of air per minute. Now, inspiratory reserve volume. Additional volume of air a person can inspire by forcible inspiration. This averages 2500 ml to 3000 ml. Now, expiratory reserve volume, ERV. Additional volume of air a person can expire by forcible expiration. This averages 1000 ml to 1100 ml. Now, residual volume of air remaining in lungs even after forcible expiration. This averages 1100 ml to 1200 ml. Now, by adding a few respiratory volumes described above, one can derive various pulmonary capacities which can be in clinical diagnosis. Now, inspiratory capacity. Total volume of air a person can inspire after normal expiration. This includes tidal volume plus inspiratory reserve volume. Now, expiratory capacity. Total volume of air person can expire after normal inspiration. This includes tidal volume plus expiratory reserve volume. Functional residual capacity. Volume of air that will remain in lungs after normal expiration. This includes ERV plus RV. Now, vital capacity. The maximum volume of air a person can breathe in after forced expiration. This includes ERV, TV and IRV, TIE. Or the maximum volume of air a person can breathe out after forced inspiration. Total lung capacity. Total volume of air accommodated in the lungs at the end of forced inspiration. This includes RV, ERV, TV and IRV और वाइटल कैपेसिटी प्लस रेसिडुअल वॉल्यूम. ठीक है, मतलब वाइटल कैपेसिटी प्लस रेसिडुअल वॉल्यूम. Vital capacity include TI plus residual volume. Now, exchange of gases. Alveoli are the primary set of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on their pressure concentration gradient. Solubility of gases as well as thickness of membrane involved in diffusion are some important factors that can affect the rate of diffusion. Pressure contributed by individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in the atmosphere, air and two sites of diffusion are given in table 17.1 and 17.3. The data given in table clearly indicates concentration gradient for oxygen from alveolar to blood and blood to tissue. अभी नीचे एक बॉक्स दिया गया, इसमें देखेंगे आप एटमॉस्फेयर एक 159 टू 0.3 रहेगा, एटमॉस्फेरिक एयर का O2 का 159, एल्वेोलर एयर 150, एटमॉस्फेरिक एयर 159, CO2 0.3. फिर याद कैसे करना है इसको, ये वाला तो रटना पड़ेगा 159 0.3. उसके बाद एल्वेोलर टू ब्लड, ब्लड टू टिश्यू ABBT. ठीक है, 140 40 40 45 104 40 40 45 95 40 40 45. ठीक है, 104 40 40 45 95 40 40 45, मतलब ब्लड और टिश्यू का सेम है. उसके बाद gradient is present for CO2 in the opposite direction, that is from tissue to blood and blood to alveolar. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance composed of thin basement membrane supporting the squamous epithelium and basement membrane surrounding the single layer endothelial cells of capillaries in between them. However, its total thickness is much less than millimeter. Therefore, all the factors in our body are favorable for diffusion of O2 from alveolar to tissue OAT out, and that from CO2 from tissue to alveolar CTA. Now, transport of gases. Blood is a medium for transport of O2 and CO2. About 97% of O2 is transported by RBC in the blood. The remaining 3% of O2 is carried and dissolved straight through plasma. Nearly 20 to 25% of CO2 is transported by RBC, whereas 70% of it is carried as bicarbonate. About 7% of CO2 is carried and dissolved straight through plasma. Now, transport of oxygen. Hemoglobin is a red color iron containing pigment present in the RBC. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2, partial pressure of CO2, hydrogen ion concentration, and the temperatures are other factors which can interfere with this binding. Sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called oxygen dissociation curve and is highly useful in study the effect of factors like PCO2, H plus concentration, and on binding of O2 with hemoglobin. In the alveoli, when there is high PO2, low PCO2, lesser H plus concentration, and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues where low PO2, high PCO2, high H plus concentration, and higher temperature exist, the conditions are favorable for dissociation of oxygen from oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissue under normal physiological conditions. Now, transport of carbon dioxide. CO2 is carried by hemoglobin as carbiminohemoglobin about 20 to 25%. This binding is related to partial pressure of CO2. PO2 is a major factor which could affect this binding. When PCO2 is high and PO2 is low as in the tissue, more binding of carbon dioxide occurs, whereas when the PCO2 is low and the PO2 is high as in the alveoli, dissociation of CO2 from carbiminohemoglobin takes place. Dissociation of CO2 from carbiminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissue is delivered at the site. RBC contain very high concentration of enzyme carbonic anhydrase and minute quantities of same is present in plasma too. This enzyme facilitates the following reaction in both the direction. CO2 plus H2O, carbonic anhydrase, फिर देगा H2CO3, कार्बोनिक अनहाइड्रेस वापस दोनों तरीके से. सिर्फ एंजाइम का नाम इसमें याद रखना, कार्बोनिक अनहाइड्रेस इसका एंजाइम रहेगा. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBC in plasma and form SCO3 minus and H plus. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction leading to formation of CO2 and H2O. Thus, CO2 trapped as a bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. Regulation of respiration. Human beings have significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissue. This is done by neural system. Specialized center present in medulla region of the brain called respiratory rhythm center. तो medulla में जो होता है वो respiratory rhythm center होता है, तो MRR याद रखना. It is primarily responsible for this regulation. Another center present in the pons region of brain called pneumonic center. pons में जो रहेगा उसको pneumonics बोलेगा, तो PP याद रख लेना. Can moderate the function of respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increases in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustment in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arc and carotid artery also can recognize changes in the CO2 and H plus concentration and send necessary signal to the rhythm center for remedial actions. The role of oxygen in regulation of respiratory rhythm is quite insignificant. अब disorders of respiratory system पढ़ेंगे. अस्थमा. Asthma is difficulty in breathing and causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major cause of this is cigarette smoking. Now, occupational respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissue, and thus cause serious lung damage. Workers in such industries should wear protective masks.